All right, turn in what you have right now. Whatever you have. Turn in whatever you have right now. Okay. Can somebody give me the process? So I've got DNA here, it's double-stranded, I know that. And then I've got this other strand of nucleic acid that's being made from DNA. What process is this, y'all? Not replication. Not translation. Transcription. So remember how last time I told you this is the diagram that almost every test wants to give you because they want you to think it's replication. But notice that even though you got a DNA molecule right here, you're making what looks like RNA because look, RNA has U, right? And it's single-stranded, so that's your clue right there. This is a single-stranded molecule with U's on it. So we are making mRNA. All right. 3 ways that RNA is different from DNA. Somebody give me one. One-stranded. One-stranded. Good. It's what? It's a what?
Outside of the nucleus.
No, it can be inside the nucleus too.
It can be outside the nucleus.
It can be. It can be both. No thymine. What else?
Adenine bond with uracil.
Say that one more time.
Adenine bond with uracil.
Okay, so no thymine. So there's uracil. Give me another one. So my saying no thymine means the same thing as has uracil. So what's another one? Look at your notes. It's like the first thing we talked about. What do your notes say? Say that one more time. One more time. No. So the first thing you wrote down has something to do with the sugar. Has something to do with the first letter. What does the D stand for? What does the R stand for? So the D stands for deoxyribose sugar. So what does the R stand for? Ribose sugar. So it has a ribose sugar. Make sure you're writing this down. if you didn't write it down before. All right. So the next one said, what would be the nucleotide sequence of the RNA that is transcribed from the DNA with the nucleotide sequence GCT, AAT, and CCG? So you guys tell me. G binds with, and then C binds with, T binds with, good. A binds with, U. A binds with, T binds with, C binds with, C binds with, and then G binds with. All right, in RNA, thymine is replaced by what? Uracil. In eukaryotic cells, the RNA is copied from the DNA in the what? No, the nucleus. Okay, so transcription occurs in the nucleus. Please correct yourself. Whatever you need to correct, go ahead and correct that now and submit your corrections. God bless you. When you're done submitting your corrections, please turn back to your notes. We briefly discussed translation. And so now, okay, so I told you guys that we're going to use mRNA in a ribosome, and we're going to put some amino acids together to make protein. Cool. We're going to do all of that in cytoplasm. But today we're going to discuss exactly how it is that we do that. Now, this concept is pretty easy, but it could be very hard if you are distracted by your cell phone. So what I need for you guys to do is put your cell phones away, like put them face down. And what's going to need to happen is you're going to have what we call self-control. Take out your pen or pencil, and you're going to have to write this down and try and focus the entire time so that you can get these concepts. Okay, so in your announcement today, so you should have already submitted your correction for the warm-up. Okay, you notice that you've got homework. You'll click on it tonight. So far, you guys have had 3 sets of homework that will come due and will be graded. So that could be 3 zeros that nobody wants. So hopefully you've been doing your homework every single day. So just so you know, that's 3 zeros that will hurt. Do your work. Just saying. This activity, you're going to click on it. Pick a monster. There's 4 monsters. It doesn't matter which one you pick. You're going to go through the transcription. Okay. So it's got a DNA sequence right here. You're going to transcribe it into RNA here. So ACC becomes what for mRNA? UGG. Right. And so you're going to do that for all the mRNA. Then you're going to use your codon chart, which I passed out last class. Make sure you've got one. I passed out a codon chart last class. It should be with your stuff. Okay, we looked at it last class. And you're going to figure out the amino acid sequence. Then you're going to that amino acid sequence here, write down all the traits for your monster, and then you're going to draw your monster and turn it in. So write down the traits, draw it, turn it in. That is homework tonight. Do we understand each other? Yes, sir.
The homework you sent us, I printed them. Is that okay?
You did what? You printed it?
Yeah.
Oh, yeah, that's fine. Okay.
Also,
Mm-hmm.
also, in the class, you guys are like teachers who can do the codon chart.
You don't have a codon chart?
No, I have it. You think I have to do this? How about this?
You don't remember how to do it? All right, yeah, we're gonna go over it again. All right, so anyways, that is homework, and yes, I will go over the codon chart again. What else? Your corrections for your exam are due this Friday. So that's tomorrow by midnight. Do your corrections if you failed. If there is any way to pass this class, if you are struggling to pass this class and you don't correct your test, that's on you, because you have that opportunity. Correct your exam, turn it in, text or email me when you do, and then I'll open your test so you can retake it. Okay, so please don't sit there and just accept the failure. Go back, correct it, and so you can at least get a 70. Anywho, so click on protein synthesis notes, and what I want you to do is I want you to start copying page 2 now. So we did page 1. I want you to copy page 2. It looks like that. So you have to click on it, because I'm quite sure that you can't read it from the screen here, because it's kind of hard to read on the screen. If you click on it, then it's on your screen. Danielle, put your cell phone away. Okay. Guys, once you're done drawing that, I need for you to take out your codon chart. Okay, so you should pretty much have this drawn by now. Just as a recap from last time, heads up. Last time we talked about what happens or how DNA is actually read and used. We know that DNA stays in the nucleus. It cannot leave. So the information in DNA has to be copied. And that process is called transcription. So we're going to copy that information and then take it out to the cytoplasm where we can actually build the protein. Now, that doesn't mean I'm copying DNA. So let me repeat myself. We're going to copy the information from DNA, take the information out to the cytoplasm, attach to ribosome, and make protein.
Oh.
So that does not mean that I'm copying DNA. DNA replication is something totally different. This is how we use DNA. So when we copy that DNA, we copy it into a molecule called mRNA, messenger RNA. Okay? We use only one enzyme, RNA polymerase. RNA polymerase is going to unzip and unwind DNA, and it's going to build the mRNA. Now, some of the things that were different was that mRNA is, first of all, it is single-stranded. It has a different sugar. That's why it's called RNA. So it's ribonucleic acid. So it's ribose is the sugar. And it doesn't have T. So that every time RNA polymerase sees an A, it's going to put a U, because RNA doesn't have T. Okay? So we only are going to copy one strand, and that one strand is called the template strand. The other strand that we don't copy is called the coding strand. Okay? That's transcription. Okay? Not replication, it's transcription. So this is when we're going to copy the information from DNA into a molecule of mRNA. And we do this because, again, DNA cannot leave the nucleus. It's stuck. But proteins have to be built in the cytoplasm. And the whole purpose for DNA is that it is instructions for building those proteins. Okay, so we've got to get the instructions from DNA. We got to copy them. It's like taking a picture of them and then taking them out of the nucleus out to the cytoplasm with the ribosome and make some protein. That is transcription. Okay? So we unzip DNA. RNA polymerase is going to build the mRNA molecule. And then we know that mRNA has U's instead of T's, so that the new mRNA molecule looks something like that. Now, we also know that DNA has junk. So please go to that part of your notes. I'm going to go over that one more time. DNA has junk. In other words, there are parts of DNA that code for nothing. They code for nothing. And because they code for nothing, they can't be used. They're just like a whole bunch of A's next to each other, a whole bunch of T's next to each other. They're pointless. But RNA polymerase doesn't know that. RNA polymerase doesn't know that that's junk. And so RNA polymerase is going to transcribe everything. It's going to copy everything. So it's almost as though you go in and you take a picture of something, and part of that something is pointless and junk. Well, you've just taken a picture of the whole thing. That's what polymerase has done. It has just taken a picture of the whole thing. So later on, that mRNA is going to get cut up. So what we're going to do is we're going to cut out the junk that we just copied. The junk is called the introns. So we're going to cut those out. And the exons are the parts that we want. So we're going to connect those together. Okay, did we draw all that? Yeah. Yes. I'm in different places in different classes, so I want to make sure that we did go over that. We did? Okay, I want to make sure. Okay, so the introns are the copies of the junk. We're going to cut those out, and we're going to glue the good stuff together, and the good stuff are the exons. So what does that look like? Here's that junk. So everybody, heads up. Here's that junk right here. You see that? That's junk. We don't want that. So what are we going to do with that junk? What are we going to do with those introns?
Remove it.
We're going to remove it. We're going to cut it out. And the rest of it is good stuff, right? So what are we going to do? We're going to paste the good stuff together. So the exons are the good stuff. Okay? Now, let's do this. Let's add to your notes this part, because some of us are still struggling with reading the codon chart. So, do me a favor. What I want you to do is I want you to write these 7 codons. Write those down right now. On that page under where you wrote exons and all that good jazz. So page 2 of transcription. And let's practice. So I want to make sure that you guys can use your codon charts. That being said, please do it now. Don't play on your cell phone. Do this now. Well, yeah, just write this down, buddy. Come on. Okay? Take out your codon charts, please. Take out your codon charts. If you do not have a codon chart, say something. Let me know. Okay. So let's look at this. Okay, so this is the mRNA. Okay, so we just transcribed it. Okay, so we got it from the DNA. Remember, transcription is just copying from DNA, right? So we just copied the information from DNA into a molecule of mRNA. So that's transcription. Now we like to put them into sets of 3. Each set of 3 is called a codon. Okay, each set of 3 is called a codon. Now this mRNA is going to leave the nucleus, go out to the cytoplasm, attach to a ribosome, and begin translation. And that's when we're actually going to build the protein. Protein has monomers of amino acids. Okay, so the parts of a protein are called amino acids, all connected together, right? So how do we figure out which codons code for which amino acids? We use our codon chart. So let's take out the codon chart and let's look at A to G, the first codon. So heads up. Here's how we use the codon chart. Here's the first letter. The second, the third. So, A, yes. U, yes. And G. Heads up, Moyette, you're still confused, remember? A, then U, right? And then the way we know which one it is, because we know it's this box, right? The way we know which one it is we look over here and it's G. So that is the amino acid. A, U, G codes for the amino acid methionine. All right, so because we're lazy and we don't want to write out methionine, because who wants to do that, we're just going to put the first 3 letters. That's the first amino acid. Next amino acid, we're going to use this codon. Again, every set of 3 is called a what? Codon. So C, G, G. First letter, C, right? Second letter is G. So we know it's this box, right? So C and G, right? And then the third letter was G. So we know it's this one right here. Wouldn't matter because they're all the same, but whatever. Right? First, second, third. You with me? So that's arginine. I'm just going to put the first 3 letters. Okay, let's do the next one. Again, what is a set of 3 mRNA nucleotides called? Codon. Next codon, UCC. U, that's the first letter, right? C, so we know it's this box, right? Okay, and the next one is C. It wouldn't matter because they're all the same anyway. So the next one is what? Serine. First 3 letters. I don't want to write the whole thing out because, I don't want to do that. Let's try the next one. GAU. G, right? First letter, G. Second letter is A. So we know it's this box, right? Third letter is U. So it has to be aspartic acid. How are we doing so far? I'm going to put ASA so it's not asparagine. Aspartic acid. Now, how do we use the second chart? Same information, just different setup. So the next, what is this called again? AAG? What is a set of 3 RNA nucleotides called? Codon. Next codon is AAG. All right, I start in the middle. A, G. What's the amino acid? Lysine. Okay, next, GGC. G in the middle, G, C. What's this amino acid? Glycine. How you doing, Moyette? You good? Last one is C, looks like CAU. C, A, U. Histidine. Oh, look, I've just made my, what I like to call a polypeptide. Now, why do we call it a polypeptide? Poly means many, yes? Okay. The bonds between amino acids, guess what? They're called peptide bonds. Poly, many, peptide. Okay? Because there's many peptide bonds. Or AKA a protein. It's the same thing. Welcome to biology. There's so many names for the exact same thing. How we doing? Awesome. All right. Now, we went over how to use the codon chart. Now I need to teach you exactly how this works. And you're not going to like it, but if you pay close attention, you'll get it. And you'll be like, this is repetitive.
On the bottom?
It says polypeptide and protein in parentheses. All right. So we kind of just went over that. What is this process? Where I take the information from DNA and I make a strand of mRNA. What is that process called? Transcription. I call that transcription. There we go. Now, what is this process when I take that mRNA, leave the nucleus, go out to the cytoplasm, and start making some protein? What is that called? Another T word. That's translation. Okay? And these little parts of protein The monomers of protein are called what? Starts with an A. Amino acids. Excellent. Okay. Now, if I have a set of 3 mRNA molecules, what is that called? A what? Codon. I feel like we drew this last class. Am I wrong? Did we draw this last class? Yes or no?
No.
So this goes under where it says, we're on page 2 of protein synthesis, and it says protein synthesis page 2 translation. Yes? We wrote some background. Yes?
Yes.
And then there was another section underneath it. Have we started that section? All right, cool. So here's what I want you to do. On that section, listen carefully. I want you to write down the DNA. I want you to write down the mRNA. And you can go ahead and write down the protein because we've already practiced that. And I will give you a minute to do that. That's where this information goes.
Can we remove the Watson covering the box?
Is Jennifer here? Is Jennifer here? Melanie, what seat number is that you're sitting at?
16.
Alexandra, what seat number is that?
17.
All right. So we kind of just went over that. If you guys would divide that up into threes, and if you divide mRNA up into threes, what are those called again? Excellent. And go ahead and label just the first codon. So codon is a block of 3 mRNA bases. Write that down. Okay, now I'm going to introduce you to the next tRNA, or the next RNA molecule. It's called the tRNA. And we're going to talk about that one. So we already did mRNA. Now mRNA is going to leave the nucleus because transcription is done. We've already cut out all the junk from the mRNA molecule. We don't need the introns. We've cut those out. We've glued together the exons. We're going to take the final mRNA molecule and we're going to let it just go ahead and flow out of the nucleus. It's going to flow out of the nucleus. So the next one that we're going to work with is the tRNA. Guys, can you write these down? Write them down under translation practice, where it says translation practice. The start codon is AUG, and there are stop codons UGA, UAA, and UAG.
Where is that?
The second section of your notes where it says translation practice. So write it under everything. The thing about these is that you're going to see them on your codon chart. Right here, sweetie. See how I wrote this? You wrote your background, and then we just wrote this up under here, right there, in that section. You're going to see on your codon chart that it says start, and it says stop. So you're going to know start and stop. So go down to you good now? All right. So under where it says tRNA, where you wrote tRNA, I want you to write this information. It looks like a clover leaf. The anticodon is at the bottom, and the amino acid is on the top. Now, I just told you that the mRNA has the codons, right? So I just told you that mRNA has codons. And so now I'm telling you that tRNA has the anticodon. What does the prefix anti mean? Opposite. So that if on the mRNA the codon is AUG, what's the opposite of that? What's the anticodon? So AUG, the opposite of AUG would be what?
GAC.
So UAC, right? So it's the opposite. So the anticodon is always the opposite of the codon. After you've written this information, under tRNA, I want you to go over to the right side where it says diagram, and I want you to draw this diagram because the rest of this is too hard to draw. So I want you to draw this diagram. Okay? This right here is where the amino acid would be.
Wait, what is that?
So after you write this information under where it says tRNA, I want you to go to the right side where you wrote diagram, and I want you to draw this. Okay? And label it. So this part right here is the anticodon. And this part right here, is the amino acid. So let me repeat myself. Transfer RNA, it's a whole new molecule. On the bottom of that molecule is the anticodon, so the opposite of whatever the mRNA codon is. And then you've got the tRNA right here, and on top is the amino acid. So make sure you draw it and label it. So the bottom is the anticodon, the middle is the tRNA, the top is the amino acid. Okay? Now go down in your notes to where you wrote ribosomes. Here's the purpose of the ribosome. This is to facilitate the coupling or the attaching of the tRNA anticodon to the mRNA codon. That is what ribosomes do. The structure of ribosome contains ribosomal RNA and proteins. There are two parts to ribosome, a large part and a small part. And then here's the picture. You should draw this one here because I find this one to be extra. So you can draw that to the right side where it says diagram. So the purpose of the ribosome is to attach the mRNA to the tRNA, to couple, attach, bind, whatever, the tRNA to the mRNA. The tRNA has the anti-codon, and the mRNA has the what? Codon. The tRNA has the anti, and the mRNA has the codon. Take a picture. All right, we're not going to go through the different sites because all I need for you to do is just label them, E, P, and A. I'm not real particularly concerned about you knowing each site of the ribosome. Nor am I particularly concerned about you knowing this part. But let's go through this. So here's your step one. And if you guys would go ahead and turn to the back of your page 2. You just drew it today. So we're past transcription, right? So we're in the cytoplasm. So you're at the top, and you've drawn the ribosome. I want you to label the E, the P, and the A. I want you to draw your mRNA and go ahead and put the codon. So the codons on the mRNA are A, UG, CUG, and AAU.
So we draw it on a separate page?
Oh, Jesus. Okay. No, let me show you a picture. So we drew this earlier, right? So we're here. Did everybody draw this when I asked you to? Okay, so we're here, right? Okay. So there, I want you to draw this.
On all of them?
Say that one more time.
On all of them?
No, just on the first one. Okay, so first things first. Here, and make sure you label everything. So first things first, I have the ribosome here, right? So it's got 2 parts, and it's got 3 binding sites, the E, the P, and the A. Yes? Here's my mRNA. Here's the codons on the mRNA. AUG is the first one. CUG is the second one. AAU. What is this molecule called right here? Can anyone tell me?
Uh,
That's a tRNA. That is a tRNA. I mean, it says it over here, but I'll write it over here too. They're both tRNA. Okay, and this is my ribosome, right? What does a tRNA look like it's doing? What has it done?
then,
It did what?
then.
It's already bent. What did it do? It bonded with the mRNA. Excellent. Now, how does it know where to bond? How does it know where to attach?
Any place that attaches the three
Codons.
three codons. Any place that attaches to three codons that has the nucleus.
Excellent. So right here I have the codon, and what is at the bottom of the tRNA? The what? What's at the bottom of the tRNA? You just drew it. What's at the bottom? If that's the codon there, what does the tRNA have? It has the anticodon. Make sure you label that. That's how it knows where to bind, right? The anticodon has to match with the codon. So A with U and C with G, right? You with me? All right. Just want to make sure everybody's on the same page. What is on the top here? What is this on the top of the tRNA? What is it called? The amino acid. Yes, we're good. Cool. All right, now we have attached to the P site. Okay, now another tRNA comes along. And guess what? What does it have on the bottom? What is that called again? The anticodon. Okay, and so what would the anticodon be? It's a CUG. C binds with, U binds with, and G binds with. So that's the anticodon. That's how it knows where to bind. All right, so another tRNA comes and binds. Yep, you gotta draw that. On the next one, you're getting it. We are so getting there. We've already labeled that, so we don't need to label it again. So a second tRNA comes along and binds. Yay! Say it one more time. I can't hear you. The things on the top? These things? They're not clouds, they're ribosomes, but yeah. So we're on the second one. We're on the second one, right? You have 4 of them. Say it again. So draw it again and make them bigger? That's step 2. All right, now we're going to do something interesting. Oh my God, what we just do?
This connected from the to the
Okay, so the first amino acid popped off and bonded to the second amino acid. Draw it. On the 3rd ribosome, y'all. On the 3rd ribosome. On the 3rd ribosome. Not a cloud. The 3rd ribosome. So yeah, you got to draw all this again. The only difference is now that methionine is now bonded to leucine. Now, these are 2 amino acids, right? We established that earlier. Proteins are made up of amino acids, right? We established that. If these are 2 amino acids, what is the bond between amino acids called? What is this bond called? Peptide. Label it. Pep and tide. So now we have just built, Ms. Johnson can't write. We have just built a peptide bond between methionine and leucine. Met and leucine. All right, now we have to move on. So here's what we're going to do. We're going to shift. What? We're going to shift. And guess what? When we shift, this tRNA right here is like pointless now because it doesn't have an amino acid anymore. We don't want it. So guess what we're going to do with it? We're going to chunk it. So draw this one. I'll take all this junk off. You guys have another ribosome, right? Yeah. I think this should be your last ribosome, if I'm not mistaken. How many ribosomes did I make you draw?
4.
This is the 4th one. So we're going to chunk that first tRNA, right? And can you guess what's going to happen next?
Okay. The new protein or protein. It's gonna do the job of it's gonna do the job of both of them.
I hear where you're going. Well, let me help you out. The next step is we're going to get a whole nother tRNA that comes along and attaches here. So this guy's getting chunked. Now we're just going to start the process all over again. A whole new tRNA. How are we going to know which tRNA to bind here? Like, how do we know which tRNA to put there? The one with the right anticodon. Gee whiz, which anticodon are we going to need to bind here? UUA. So whatever tRNA has the anticodon UUA is going to bind right there. And we're going to do the whole thing all over again until we get one really long polypeptide and we're all done, and we can wrap it all up and call it a protein, and it goes and does its job. But that's how we add amino acid to amino acid to amino acid to amino acid in that process. Say that one more time. This one goes on the fourth one, I believe. We're on the fourth one, right?
We finished this one.
Right, this was the fourth one, right?
Oh.
Wasn't this the fourth one?
Yeah. Yeah, I just didn't know. So, are we doing the water zone?
Say that one more time.
Are we doing the water zone?
No, we're going to add a tRNA in just a minute to the fourth one. Okay, so you can draw yourself a tRNA that's kind of coming in here. Just like that. And we know that the anticodon is going to be U, what? A. And then we just keep going in that fashion until we build an entire polypeptide. And what does poly mean again? Many. And then the peptide is the what? That's the bond. And so polypeptide is just one big protein. Okay? And ultimately, this is the end. That polypeptide. All right. Turn those notes in. Next class, because we didn't get to it this class, next class we'll do our lab, and then we'll talk about mutations as well. So we'll do both for next class. Oh, speaking of labs, make sure you turned in your DNA structure lab. Remember when we built DNA? You just had to take a picture of it. Let's see. That wasn't last class, but it was the class before. So the one where we built the structure of DNA with Candy, remember that? So all I wanted you guys to do is put your picture right here and label it using text boxes.
Wait, did you submit it right here?
Yes.
Did you just submit it right?
Yes.
Oh.
So how many of these, how much pages are we?
When we're all done with our notes, you will end up, looks like one, two. So your submission should be like 3 to 4 submissions. And so I should see DNA structure, right? We did properties of DNA, replication. We did transcription. We just finished protein synthesis, right? I should see all of that all in one place. So it doesn't matter when you say resubmit, I see everything. So if you never submit the first set of notes, I'll see that. So submit all your notes. I can see all of them. All I have to do is like click the little down arrow and it'll show me everything you turned in. Now we never got to the replication lab, which is fine. So all I want you to do is post your Candy DNA and make sure you label it. Just take a picture and then paste it there. And when you turn it in, you just hit share. Make sure it's editable. Copy and turn it in. We've done that several times. You guys should know how to do that by now.
Wait, are you talking about a library building?
Yeah, with the candy?
Yeah.
Yeah, that's what I'm talking about.
But I have a question.
Yes, ma'am.
Which one? What is the cell energy? What is it?
So if you're missing something and you don't know what it is, look at the screen. Go to announcements. Everything is here. So that's somewhere probably way long ago. It's not that. So I would say late October. You'd have to find it in the announcements. But it is there.
Yeah.
So let's see. Look, it's right here. It's from Thursday the 29th through Friday the 30th. And so if you click on it, it'll pop it up. And there's the assignment. And open it up. And that's what we did that day. That's what you need to do when you're absent to make up your work.
We just keep these books.
Thank you.
Mm-hmm. So what?
Just keep these books.
Yeah, because we never got to it. Here's what I need you to do. Do you guys think you could put your gloves in your backpack and not forget them on Monday?
No.
No? Okay, do you need to put them back in the box? Because we can do that. So on Monday, we will do the candy lab together. We'll do it together, slowly, so that we know exactly what we're doing.
The same one we did last time?
It will not be the same one. Yeah. Okay, here's what you should do. You can access things on your cell phone and turn it into things on your cell phone.
The project report, it ends on, before, good and break, right?
Say that one more time.
The progress report, when is it?
Progress report. Probably next Friday. Because what we started last week. This is the second one. So yeah, next Friday is the third. Yes, sir.
Okay, so what was that question? Like, I was looking through it, we're just learning.
Okay, hold on a sec.